Find evidence for your research with Europe PMC. Current research workflows can be quite complex. In addition to advanced tools and methods used at the bench, information research becomes more and more sophisticated. It used to be all about literature, reading reviews and papers to stay on top of your field. Nowadays, in addition to journal articles, you will find important scientific information present in the form of various research data, chemical structures, DNA sequences, genomic variants, expression profiles, or protein interaction networks. Although these types of data comprise an important part of biological knowledge, they are often difficult to interpret directly by users. Such diversity in the data formats makes it difficult to integrate them into a coherent workflow. This means that researchers often have to operate several resources at the same time. Comparing sequence variants from healthy and diseased individuals, while also looking at the protein structure to see how mutations would influence protein folding, checking with a small molecule database to see if ligand binding would be disrupted, and perhaps scrolling through a review describing the disease symptoms to see which other organs and systems may be affected by this condition. At Europe PMC, it is part of our mission to create solutions that make it easier for scientists to navigate the data-rich literature in the most efficient way. We have created SciLight, a tool that highlights biological terms or relations such as diseases, chemicals, or protein interactions on abstracts and full-text articles. These terms are identified by text mining algorithms developed by a variety of text mining groups. When reading a paper, on your right-hand side you will notice a menu titled Show Annotations in this abstract. By checking the corresponding boxes, you can select different concepts that you would like to be highlighted in the text. SciLight annotations include core-named entities such as gene and protein names, organisms, diseases, chemicals, and gene ontology terms. For those unfamiliar with gene ontology, it is a controlled vocabulary that defines concepts used to describe gene function. Membrane, transcription, catalysis, those are some examples of gene ontology or GO terms. In addition to these named entities, SciLight also provides annotations for biological functions, such as gene functions or gene rifts, as well as biological events, for example, protein phosphorylation, and functional relations represented by gene-disease associations and protein-protein interactions. One way in which SciLight can be helpful for your literature research is helping you to scan the article for key concepts, to understand if it is relevant to your topic, and to skim read the content, focusing on the important bits without having to read the entire paper. You can see what are the main players described in the text, which model organism the work was done in, or the kinds of drugs that were used for the treatment. For example, in this publication, you can directly see that the results presented have been done in different model organisms, including rabbits and mice, as well as human brain samples. You can see a list of individual terms found most frequently in the text to help you get a better overview. To get to the list, click on the right arrow next to the annotation type. A selection of individual terms will appear together with up-down navigation buttons, which allow you to jump to selected terms in the text. You will notice that some of the terms could be listed several times. For example, mycobacteria, Mycobacterium tuberculosis, and M. tuberculosis may be referring to the same organism. However, the text mining algorithm will list them as separate entities. Nonetheless, the list can give you an idea of the most common concepts appearing in the text. However, the main function of SciLight is linking information from scientific publications to structured resources, such as public life sciences and chemistry databases. Let's see an example of this by clicking on one of the highlighted terms. It will bring up a pop-up menu that displays a link to related database record. For example, when clicking on the highlighted concept for methoxymucolic acids, a chemical molecule, you will see a link to CHEBI, or Chemical Entities of Biological Interest Dictionary of Molecular Entities. If you follow that link, you will see a summary dedicated to this particular compound. You can find the chemical formula, mass, and charge, as well as some functional information here. Similarly, if you open a pop-up for genes and proteins, you will see a link to UniProt, a knowledge base of protein sequence and functional information. These links from text to structured resources create a knowledge map of what is known about biological objects. If you're exploring a new topic, such links provide you with encyclopedic coverage that comes from reliable public data sources. You may not be familiar with the resources commonly used in the field, so you can rely on SciLight to guide you to a trusted, high-quality database. Moreover, SciLight can help you find evidence and formulate or refine your hypothesis. As we have already mentioned, SciLight annotations include functional relations such as gene-disease associations and protein-protein interactions. For those annotation types, an entire sentence that contains the evidence for this relation is highlighted. Here is one example. In this case, we find the evidence that MKX2-5 interacts with TBX5. This annotation comes from INTACT database, which is manually curated by experienced researchers. You can see the annotation source on the pop-up menu. In the case of gene-disease associations, they can be provided by one of the two sources, DisGenet and OpenTargets. For these kind of annotations, you will find two links displayed on the pop-up menu, for the gene and for the condition. In some cases, a sentence will contain overlapping annotations. If that happens, SciLight will highlight the longest annotation, and the individual annotations within the phrase will appear in the pop-up window together. You can interchange between them by clicking on the annotation name in the pop-up. Although text mining algorithms are getting better and better at getting the most meaningful and useful information, it can be hard to discern terms due to similar names or abbreviations. For example, peanut can refer both to a plant species and Drosophila gene name. Or the acronym APC can correspond to one of the following concepts: antigen-presenting cells, activated protein C, anaphase promoting complex, or argon plasma coagulation. Sometimes, despite stringent quality checks, you may notice an annotation that is too generic or incorrect. It is of critical importance that readers find the annotations useful, so you can provide feedback on each annotation. To do so, use the feedback link in the pop-up window. You can also endorse annotations using the feedback link if they are useful to you. This information is fed back to the Europe PMC team and will be acted upon, helping to improve the annotations overall. Finally, there is one more function that SciLight can provide to support you in your research. It enables scientists to locate the primary data from the text. For instance, if you would like to retrieve and use a public dataset to cross-reference a result, or test your hypothesis in silico by using public omics data, you can focus on accession numbers in the text that link to that primary data. Accession numbers are unique identifiers given to a data record to track it in a single data repository. Such identifiers are crucial, especially because protein and gene names are often ambiguous. Accession numbers are yet another type of SciLite annotations. They're especially useful because they allow you to quickly get to the data mentioned in the paper, be it protein structure coordinates or sequencing results for biopsy samples. Here you can find the reference to PFAM record for a protein domain in the text. SciLite tracks accession numbers for several different resources, including ArrayExpress, UniProt, PRIDE, the Proteomics Identifications Database, Protein Data Bank Europe, European Nucleotide Archive, and many more. You can see a full list here. If you click on the highlighted accession number identified by SciLite, you will get an already familiar pop-up menu with a link to the corresponding data source, where you can view, download, or analyze the dataset in question. It can be difficult to make sense of the raw data in its natural form, so we experiment with data visualization techniques to help you digest information efficiently. For instance, SciLite displays a 3D view of the protein structure on the pop-up menu for datasets deposited in Protein Data Bank, or PDBE. This acts as a preview to the actual dataset, allowing you to assess the structure in its original context and get a visual cue for a wordly description. You can then continue reading the paper or proceed to the database to work with the structural data. When using SciLite tool, you will notice that the full text of some articles does not have annotations menu apparent on the right-hand side. This is because the use of the material is subject to the copyright and related license terms of the respective authors or publishers. SciLite annotations can only appear on full-text articles with a CC BY, CC BY NC, or CC0 license. However, we do text mining for the entire full-text corpus and display results in a separate tab titled Bioentities. On the abstract of the full-text page, you will see the Bioentities tab. If you click on the tab, you will be able to browse a list of concepts and entities that is similar to the list of SciLite annotations. Clicking on the name of the bioentity takes you to the corresponding database. Note that Bioentities tab and SciLite link to different resources. This is because the focus of the Bioentities tab is on individual text-mined terms rather than the primary database record. Clicking on the search icon next to the term initiates a search on EuropePMC for that term. You will also find links to the datasets mentioned within the article. Data citations in the Bioentities tab are based on accession numbers, which you have already encountered when using SciLite. In addition to the data cited in the publication, you can also see database records that have cited the article. When datasets are submitted or curated, references to the literature are frequently added, and those references are made available via the Bioentities tab as database citations. Both types of citations can be explored via the Advanced Search form. On the Advanced Search page, select Data Links and Data Citations. You can see all available options in the drop-down menu. This data search functionality distinguishes EuropePMC from other resources. Most databases will provide links to underlying primary literature, but these links might be scarce, let alone extensive. You can easily search for data associated with certain concepts or keywords and create complex data-driven search queries across multiple resources using Advanced Search from EuropePMC. For example, you can look up which publications on ulcers have a link to clinical trials, which articles on Arabidopsis thaliana provide associated mass spec data, or which papers provide both structural and functional information for protein domains implicated in unfolded protein response signaling pathway. Now you know how to search for and identify primary research data in scientific publications. However, there may be several different types of data from a variety of resources associated with a single paper. For instance, this publication is citing a protein structure from PTBE, several genomic sequences provided by ENA, and an RNA sequence from NCBI nucleotide database. In addition, it also has several supplemental data files. To explore all data associated with a particular paper, you can view its BioStudies record. BioStudies is a bento box for data that packages all data about a study in one location. You can access the link to the BioStudies record from the abstract page of the paper. Note that you will see links to supplemental files as well as links to cited data in this record. With this, we have hopefully covered all different ways in which you can explore the connection between scientific literature and research data. For more information, you can refer to EuropePMC help pages or contact EuropePMC help desk. If you're interested in the mechanism behind SciLight application, you can follow the link on the slide to the software tool article.